Well, good morning, and I hope that this presentation finds everybody safe and well as we all still try to become accustomed to the changes to our own personal lives and those of our work. So we greatly appreciate you being here for this presentation and joining us. Just to give you a quick intro to who we are, I am Vince Shire. I'm a partner at Enablement and have been practicing patent law for just under 20 years as a member of our firm's chemical patent prosecution and PTAB proceedings practice groups.
And I'm Grace Kim. Like Vince, I'm a member of the firm's chemical patent prosecution group where I'm a senior associate with 8 years of experience. Today, we will be discussing developing enablement law and its impact on pharma. If you have any questions, please enter them into the Q&A box, and we will answer the questions either during or at the end of the presentation.
So whether pre-AIA or following the AIA, 35 U.S.C. 112 contains 3 requirements: the written description of the invention, the manner and process of making and using the invention, or enablement, and the best mode contemplated by the inventor of carrying out the invention. While the AIA diminished the practical importance of the best mode requirement, written description and enablement requirements are core patent requirements and are frequently at the heart of patent prosecution contests and invalidity contentions in litigation, especially in the life sciences area.
The written description requirement and the enablement requirement are often confused with each other. Could you provide a basic process to use to distinguish these requirements?
Yes, it's important to note that written description and enablement are actually separate requirements. Written description has the focus of the inventor and is a question of possession. The question is, was the inventor in possession of the invention at the time the application was filed? The enablement requirement, on the other hand, is a focus on the public, where the question is whether the person of ordinary skill can make and use the claimed invention based on the disclosure in the application. So in order to be enabling under 112, a patent application must sufficiently disclose an invention to enable those skilled in the art to make and use it. The specification need not disclose what is well known in the art, but unless the information is well known, the application itself must contain that information.
Well, how do we assess whether or not a patent is enabled?
Enablement turns on the question of whether the specification provides enough details to permit the skilled artisan to practice the full scope of the invention without undue experimentation. To address the question of undue experimentation, the Federal Circuit established an 8-factor test in re Wands. Ironically, Wands is a case involving monoclonal antibodies, where the Federal Circuit concluded that methods to generate and screen antibodies against specific antigens were well known and routine. But the analysis of whether a claim is enabled must consider these factors and answer the question if, on balance, the totality of information allows the skilled artisan to practice the full scope of the claims.
Vince, since the ultimate legal conclusion of enablement rests on the facts, what kind of facts would be reviewed, particularly in pharma and chemical patents?
Well, one important element that is reviewed is the unpredictability factors. For example, in re Marzocchi, the court held that unpredictability of chemical reactions may be enough to create a reasonable doubt as to whether disclosure enables the claim. Most chemical reactions and physiological activity involve unpredictable factors, and the scope of enablement varies inversely with the degree of unpredictability. Another analysis related to the unpredictability is whether there is inconsistencies in the data that's presented in the specification.
In pharma patents, antibody claims have been under scrutiny and attack at the Federal Circuit. Why is this?
In part, this is due to the importance of antibodies in modern therapeutics. Antibodies are proteins produced in nature by the immune system and used frequently in biological therapies. Due to their ability to bind, alter signaling pathways, and as a result, they're involved in various diseases such as cancer, infections, and inflammation. Antibodies recognize particular target antigens, or target agents called antigens, which are bound via variable domains at the end of the antibody. The specific portion of the antibody that is bound to the antigen component is referred to as the epitope. Two antibodies may bind the same or a similar epitope and compete for binding, meaning that the presence of the first antibody bound to the antigen would prevent the second antibody from accessing and binding the epitope on the same antigen. It's been about 3 decades now since the first monoclonal antibodies were approved by the US FDA, and during this time, antibody engineering has dramatically evolved, and as a result, we've seen a tremendous explosion in the patent activity in the area of antibodies. To emphasize the importance of antibodies in the therapeutic aspects, they've become the predominant class of new drugs that have been developed in recent years. Just looking back over the last few years, back in 2018, 8 of the top 10 best-selling drugs worldwide were biologics. The global market for therapeutic monoclonal antibodies was estimated about $115 billion in 2018 and is expected to generate nearly $300 billion by 2025. And so as we see the evolution of the importance of these therapeutic weapon in their arsenal, we also see, of course, the importance of protecting the antibodies via patent protection. So just to give an example of what an antibody claim would have looked like 10 years ago, which is very common, is the specification just generally describes the use of the antibody. And in this example that I have pulled is just the Shemeron R, which is useful for diagnosis and immunotherapy related to HIV 1 and 2. And with this general description, a claim would typically be presented, which is drawn to, in this case, an isolated monoclonal antibody, or the binding portion of the antibody to the antigen, and it would define a specific function or activity, which is binding to the epitope being sec ID number 61. And it would define expressly what the antibody's activity was, binding of Shemeron to Shemeron R. The question is whether such a claim would actually pass muster as with the evolution of case law relating to Section 112.
So the first assault on antibody claims was through the written description requirement. In 2008, the USPTO issued a guidance named the Newly Characterized Antigen Test. Here's a visual chart that we'll return to again later, showing the developments in law at the Federal Circuit regarding antibody patents. So the USPTO previously provided an exception to the general written description rule for antibody claims. This allowed written description support for a claim to a genus of antibodies through the disclosure of a newly characterized antigen, as long as the production of such antibodies was conventional or routine. The newly characterized antigen test thus allowed patent owners to obtain claims covering any potential competing products that bind to the same epitope or antigen. Before the AbbVie v. Janssen case in 2014, the majority of antibody patents invalidated for lack of written description involved claims that were clearly not possessed. For example, a humanized antibody claim that was later added in a case filed before humanized antibodies were even in existence. Claim 29 of AbbVie's patent is directed to a neutralizing isolated human antibody or antigen binding portion thereof that binds to human IL-12 and dissociates from human IL-12 with a certain cost rate constant. AbbVie developed its IL-12 antibodies using phage display, which involved creating a large library of human DNA fragments and screening for those fragments that encoded an antibody fragment with IL-12 binding affinity. However, AbbVie had isolated only one primary clone, Jo9, from which several derivatives via amino acid mutations were formed. Here, AbbVie's generic claim can be classified as a set of human antibodies defined functionally by their high affinity and neutralizing activity to the human IL-12 antigen. AbbVie's patent described only one type of structurally similar antibodies rather than antibodies representative of the full scope of the genus. There was no disclosure of any structural features common to the members of that genus. So in this case, the court discussed that functionally defined genus claims can be inherently vulnerable to invalidity challenge for lack of written description support, especially in fields that are highly unpredictable, where it is difficult to establish that correlation between the structure and the function for the whole genus, and where it's difficult to predict what would be covered by the functionally claimed genus. Coming back to our timeline chart, after AbbVie v. Janssen, the USPTO still maintained the 2008 guidance. However, the examiners did begin to take the position that any functional antigen-based antibody claim lacks written description. Then came the Amgen v. Sanofi case in 2017 at the Federal Circuit. Claim 1 of Amgen's patent is directed to an isolated monoclonal antibody wherein when bound to PCSK9, the monoclonal antibody binds to at least one of some listed residues where the antibody blocks binding of PCSK9 to LDLR. In the specification, 3,000 human monoclonal antibodies were rescreened for binding to the wild-type PCSK9 to confirm stability, which were then narrowed to about 85 antibodies that blocked the interaction between PCSK9 and the low-density lipoprotein receptor, LDLR, at greater than 90%. The jury instructions given in this case basically follow the newly characterized antigen test of the USPTO's 2008 guidance, stating, in the case of a claim to antibodies, the correlation between structure and function may also be satisfied by the disclosure of a newly characterized antigen by its structure, formula, chemical name, or physical properties, as long as the level of skill and knowledge in the art of antibodies at the time of filing was that the production of antibodies against the antigen was conventional or routine. Sanofi, though, argued that the disclosure of an antigen cannot satisfy the written description requirement for a claim to an antibody. In this case, the Federal Circuit actually did not issue a final decision on the merits of written description, but remanded the case for further consideration by the district court. The Federal Circuit disparaged the newly characterized antigen test as allowing patentees to claim antibodies by describing something that is not the actual invention, the antigen. So, due to the Amgen v. Sanofi case, the USPTO issued a memorandum in February of 2018 called the Clarification of Written Description Guidance for Claims Drawn to Antibodies and Status of 2008 Training Materials. This memorandum instructed the examiners that adequate written description of a newly characterized antigen alone should not be considered an adequate written description of a claimed antibody to that antigen, even when the preparation of the antibody is known as routine and conventional. The written description must be provided for the antibody itself, and there should be some focus on the structure, such as, is the inventor aware of the actual structure-functional relationship, and are there represented number of species disclosed, or at least common structural features among the species to support a broader claim?
Grace, as we discussed earlier, there's often a blurred line between written description and enablement. These cases so far show how the courts are reining in antibody claims through the written description requirement. But isn't the focus of the courts in AbbVie and Amgen related to written description, and doesn't that also relate to the factors for enablement?
Absolutely. As is often the case, written description and enablement go hand in hand. In fact, an assault would not be complete without bringing the two prongs of Section 112 into play. One important case on enablement of antibody patents is Morphosis v. Janssen at the District Court in Delaware. Morphosis's patent was drafted in a manner not uncommon for antibody patents. Rather than claiming the antibodies by their structure, such as a sequence of amino acids, the antibodies were claimed by their function. As a result, the claims encompass a very large number of different unique antibodies on the order of at least millions. The specification only disclosed 4 example embodiments, however, that fell within the scope of the claims. Here, Janssen asserted that the claims were invalid as not sufficiently enabled or lacking written description. And the court held that the claims are not sufficiently enabled because they encompass a very large number of different unique antibodies, and where the specification only disclosed 4 example embodiments that fell within the scope of the claims. Morphosis v. Janssen shows us that when evaluating whether the specification enables the claims, there are 2 key factors to consider. The first is undue experimentation. For the full scope of a claim does not mean that the specification must identify every antibody covered by the claims, but a sufficient representative number of species is still necessary to enable the claims. The full scope of a claim covering an antibody will not be enabled if there exists an embodiment within the claim scope that cannot be discovered without undue experimentation. One example of focus in this case is non-conservative variants, which are variants of antibodies made through substitutions within the complementary determining regions, CDRs. This case held that unlike for conservative variants, which are made through substitutions within the framework regions and are unlikely to have a large impact on the antibody's functional properties, screening non-conservative variants for efficacy, such as binding properties, would require extremely laborious experimentation. Therefore, if non-conservative variants are encompassed within the claim scope, yet yield no predictable efficacy without undue experimentation, then the full scope of the claim cannot be enabled.
Wasn't Janssen's antibody itself a non-conservative variant of the exemplary embodiments or antibodies of Morphosis' patent?
Yes, Janssen's daratumumab antibody was itself a non-conservative variant of the example antibodies of Morphosis' patent. That fact may have been a contributing factor and may become a factor or presumption for judging enablement of antibody patents that are claimed functionally.
Well, Grace, also didn't the courts hold in Angstad that the presence of inoperative embodiments within the scope of the claim does not necessarily render a claim non-enabled?
Yes, but the inquiry here is very different from the cases that permit the claim to include limited non-operative embodiments without running afoul of the Section 112 requirements. The situation in Morphosis is more similar to Atlas powder, where claims reading on significant numbers of inoperative embodiments would render claims non-enabled when the specification does not clearly identify the operative embodiments, and undue experimentation is involved in determining those that are actually operative, particularly in an unpredictable art. Here the issue is that the person of ordinary skill cannot predict the function of non-conservative variant antibodies from their sequences. In fact, several of Morphosis' witnesses admitted that this particular antibody's binding properties cannot be predicted from its sequence.
Well, let's return to Amgen, just as the Federal Circuit will be. The Federal Circuit will be deciding whether 2 of Amgen patents meet the enablement requirements under 112. The claims of Amgen patents claim an entire genus. The dispute between Amgen and Sanofi involves patents for a class of cholesterol drugs called PCSK9 inhibitors. Sanofi conceded to infringement in 2014, and thus the question and dispute remaining is whether the patents are valid. Sanofi contends that Amgen must limit its claims to specific species within the genus. Amgen, however, contends that it may claim a broad genus of antibodies without having to identify every one. On August 28 of this past year, the District Court of Delaware granted Sanofi's motion for summary judgment as a matter of law for lack of enablement. In so doing, Judge Andrews held that it would take undue experimentation to practice the claimed invention because the patent claims a vast class of antibodies. Amgen has now appealed to the Federal Circuit, who will decide this case for the second time. At the center of the dispute in Amgen is what the scope of the claimed antibodies actually is. In particular, Amgen believed that the scope is only about 400 distinct antibodies and that 26 specifically identified antibodies in the patents is sufficient to enable the full scope of the claims by providing a representative number of species. Sanofi, in contrast, contends that the genus is much broader and embraces millions of distinct antibodies. Clearly, there's very, very different interpretations as to the scope of the claims at issue. Now, given the way the Federal Circuit has recently been viewing claim scope with a very skeptical eye, I believe the reasonable money on appeal is that the claims at issue will be held to lack written description and enablement. So, with respect to enablement for antibodies and where we currently stand is quite an interesting mix. The USPTO has issued patents directed to a broader genus. In fact, the struggle here is that for the years prior to Amgen, and certainly before AbbVie, the USPTO issued many such broad antibody patents like the one that we showed very early on. These are the very patents that are now covering therapeutic antibodies that are currently on the market or undergoing regulatory review. Also, many of the pending applications were drafted prior to Amgen, and as such were drafted with a very different mindset of what was patentable.
Vince, the problem that we are facing and the companies are particularly facing with inventions and the science related to antibodies is that it is very time-consuming.
Yes, and there's a very delicate balance and a race as to whether it is better to patent something very broad and quickly before someone else, to patent something perhaps narrow that was already done in the laboratory, or to wait and complete further experiments prior to filing a patent application, which would delay ultimately the filing date.
Many companies are filing amicus briefs either supporting Amgen's or Sanofi's positions in the case, as a decision could also affect their own patents and their business. Could you provide us with some insight on these third parties?
Sure, and I think it's also very important to note that looking at cases that are evolving on genus patents in general, the Federal Circuit has been taking a very close look with a very skeptical eye in terms of enablement. And so they're certainly considering the scope of the claims at issue here that are covering very important antibodies. These patents tend to be very broad. And so there's obviously a lot of interest in the pharmaceutical industry, either for or against such claims. For example, BMS and Merck have taken the side of Amgen. They contend that the first step of developing an antibody-based drug is identification of molecular structure that the antibody binds to. That would be the target antigen, or more precisely, even the epitope. And then ultimately, tying that to a disease is the most difficult part of the process. So BMS and Merck have argued that once the target is identified, it's easy to screen and create variants of the antibody that would meet the functional requirements. In fact, that argument is more akin to where we started with the In re Wan's case with respect to undue experimentation, where the court had previously stated that the methods of screening and generating antibodies was well known and routine. However, in contrast, Eli Lilly and Pfizer have taken the side of Sanofi, arguing that a purely functional claim would preempt further invention.
What is your advice for patent claiming, considering the current developments of case law regarding enablement at the Federal Circuit?
Well, I definitely think it's important to claim inventions in several ways, just to make sure you cover all your bases. Include broad claims to try and obtain the broadest scope of patent protection, but also be sure to include claims that are directed to species that are particularly described and exemplified in the disclosure.
I think it would also be a good strategy to continue filing continuation applications to keep the case pending, in case the patents become involved in litigation or IPRs or PGRs, so that the opportunity is still open to further tailor the claims to avoid the issues presented during those post-issuance proceedings.
Absolutely. As we've seen even in the area of 101, the case law seems to evolve quite quickly. And maintaining continuation applications, especially in areas of technology that have such value as therapeutic antibodies, is obviously very important to provide flexibility as well for the development of case law. But as a final note, it will definitely be interesting to see the decision in the Amgen-Sanofi case. In particular, is because many companies are currently involved in COVID-19 research and developing therapeutic antibodies for this purpose.
It's possible that maintaining the validity of Amgen's patent to keep protection for such a broad claim that excludes all other competition could discourage others from spending its resources and can preempt future inventions.
Yeah, that's always a possibility, and we're certainly at a time where things are uncertain. And depending on pharmaceutical companies to provide ways for us to combat COVID-19 pandemic. So the decision in Amgen will certainly affect patent rights, not just of Amgen, but perhaps of many other companies. But as we've seen time and time again, the pharmaceutical companies will still continue to work hard to find treatments. The question is whether their patenting strategies evolve as well.
Well, what strategies would applicants consider, or should applicants consider, when filing applications drawn to antibodies?
Well, of course, when drafting the application, it's always best to disclose as many antibodies as possible to represent the structural diversity of various genus or subgenus. In many cases now, we're able to obtain three-dimensional structures. If we can include some of the three-dimensional structure in the description, it would also be best if we could do so with the antigen bound, especially if it allows us to show the epitope and map the points of contact between the antibody and the epitope of interest. It's, of course, important to include as many working examples as we can that would show how to predict from an antibody sequence whether it would exhibit the claimed properties or binding characteristics. Ultimately, the experimentation necessary to enable the full scope of the claims, the goal would be to have sufficient information and disclosure in the application so that we minimize the amount of time and effort that the skilled artisan picking up the application would have to involve themselves in. We also have to be reasonable when we look at the scope of our claims. We should avoid having the number of antibodies within the scope of the claims being so large as to arguably embrace millions of potential variants. And to the extent possible, we should be able to use intelligent substitution strategies rather than trial and error. So in addition to broad genus claims, claims should also be drafted that are as narrow and directed as possible, even to the point that we envision picture claims that cover the specific embodiments disclosed. The number of antibodies needed to be tested to determine whether they're functional and fall within the scope of the claim should also be kept as small as possible. In other words, thinking about the issues that were raised with the non-conservative variants, we need to try to be able to establish some way of classifying and characterizing not just the points of possible mutation, but the nature of those mutations as well.
Antibodies aren't the only area where pharma has to be wary. However, it appears that the courts have turned to enablement recently in other contexts as well.
Yeah, that's absolutely correct. One of these areas is Markush claiming. Markush claiming has become a favorite tool for a large sector of the chemical, life sciences, and the pharma fields. In fact, a study that I came across back in 2014 showed that between 1994 and 2013, 1 in every 8 patents contained a Markush claim. And in fact, it was estimated in those patents that the number of structures that were covered in these Markush groups could range between 10 to the 10th to 10 to the 100th structures. So early this year, the Federal Circuit reminded us of the importance of the precision of language when we deal with Markush claims. In another Amgen case, this is Amgen versus Amneal, Amgen court provided guidance on construing claims that recite Markush groups. In particular, the concern here was the combination of the open transitional phrase comprising following the preamble of a pharmaceutical composition and the use of consisting of in the Markush groups that appear in the abbreviated elements B, C, and D of the claim shown in this slide. The court clarified that additional unrecited members of the Markush group are allowed when the recited group is preceded by comprising and include qualifying language such as at least one. The key issue was with respect to infringement analysis, and it asked whether the binders shown in subsection C and the disintegrants shown in subsection D of Claim 1 included those unrecited elements by virtue of the preceding preambulatory transitional phrase of comprising. The district court held that the Markush groups are closed to unrecited binders and disintegrants, relying on the traditional Markush case law. Amgen then appealed and contended that Claim 1 is open-ended because of the transitional phrase comprising at the beginning of the claim. Amgen further argued that since the consisting of terms in subsections C and D are preceded by at least one, the Markush groups only limit the at least one binder or disintegrant. In other words, the comprising language permits the inclusion of additional binders and disintegrants as long as the composition includes at least one binder and one disintegrant that are selected from each of the respective Markush groups. So the Federal Circuit agreed with Amgen, and in so doing, they distinguished over Multilayer and Shire, which construed the term consisting of as limiting a claim element to members within the recited Markush group. So this decision clarifies that additional unrecited elements are allowed when the limited claim elements are preceded by the open phrase comprising and include the qualifying language to the Markush group, such as at least one.
So in addition to transitional phrases and how they may raise scope issues, the courts also recently looked at the enablement of broad claims, which will have a direct impact upon Markush claiming in the Idenix case. So this is the Idenix v. Gilead case. The nucleosides claimed in the 597 patent contain a sugar ring having 5 carbon atoms, numbers 1' to 5', as well as a base. At each carbon, substituent atoms or groups of atoms can be added in either the up or the down position. Idenix argued that the key to its invention and to the treatment of hepatitis B virus is the use of 2-methyl-up nucleosides. Nucleosides having a methyl substitution at the 2-up position of the molecule sugar ring as illustrated in the structure on the right. Gilead argues that the characterization is overly broad, as the patent provides no guidance in determining which of the billions of potential 2-methyl-up nucleosides are effective in treating hep C virus. According to Gilead, the patent primarily describes 2-methyl-up nucleosides that have a hydroxyl group at the 2-down position. But Gilead's accused product has fluorine, not hydroxyl group, at the 2-down position. The specification contained 18 formulas, each of which is represented by a diagram with variables at multiple positions. One of the embodiments disclosed in the specification is shown on this slide. The 2-up position in this formula, represented as R6, includes a methyl group as one of 24 possible substituents. The court held that even if this formula is limited only to its 2-methyl-up variations, the formula provides more than a dozen independent options for the R1 position, at the 2-down position, at the 3-down position, and multiple independent options for the base. The district court held that this formula alone discloses more than 7,000 unique configurations of 2-methyl-up nucleosides. The amount of testing that would be required for the compounds in the specification alone for efficacy against hep C virus weighs in favor of non-enablement. So the Federal Circuit held that the listing of substituents was not enabled and also lacked written description because it encompassed many thousands of compounds, making the listing almost essentially unlimited. The Federal Circuit agreed with the district court's calculation that the formula alone discloses more than 7,000 unique configurations of 2-methyl-up nucleosides, and thus reached the same conclusion that the amount of testing required for the compounds in the specification alone for efficacy against the hep C virus weighs in favor of non-enablement. The court also held that it would be improper to rely on a person of ordinary skill in the arts knowledge to meet the enablement requirement. Now, this is an important point in this case. Even though Idenix argued that a person of ordinary skill would understand the focus of the claim to be inhibition of the NS5B polymerase to effectively cure hep C virus, and would know which candidates were likely to inhibit NS5B and would test only those, resulting in a predictable and perhaps manageable group of candidate compounds, the court held that it would be improper to rely on a person of ordinary skill in the arts knowledge of NS5B to fill in the gaps in the specification. They held that it's the specification, not the knowledge of a person of ordinary skill, to supply the novel aspects of an invention in order to satisfy enablement.
Well, Grace, this decision really should not be much of a surprise. Wyatt and Cordes versus Abbott Labs back in 2013 is a very similar case in which the patents at issue had a claim encompassing millions of compounds made by varying the substituent groups, whereas only a small portion of those would actually have the claimed functional effects. In fact, reading the Idenix case, this was very important in their analysis. In the eyes of the Federal Circuit, because they found the patent invalid for non-enablement in Wyatt, they must also do so here, too.
Is it important that Idenix failed to make a compound that actually worked for treating hep C?
Sure, I think that is a factor as well, as is the fact that Gilead was the one who did develop the compound and blockbuster drug that treated hep C, which was encompassed by Idenix claim. Ultimately, Idenix missed out on a multi-billion-dollar drug. Also worth noting in Idenix is that the court also found a lack of written description in this case, determining that there were insufficient blaze marks to direct the person of ordinary skill in the art to a specific subset of 2-methyl-up nucleosides that would be effective in treating HCV. So Amgen versus Neal provides a cautionary tale of issues that may arise using multiple phrases throughout a claim. The use of Markush language raises a rebuttable presumption of a closed group, but as we found in the Amgen versus Neal case, when we use qualifying words such as at least one, it may lead to some very problematic consequences. However, this is very common language that is used in many, many different patent applications. And often the intent of the drafter is to permit more than one member of the Markush group. But following Amgen versus Neal, we really need to take a really close look at how we use multiple transitional phrases in claims, what the consequences in the interpretation for infringement and even in 112 may be. And we need to consider alternative language that makes it clear within the claim where perhaps, as in Amgen, maybe we only wanted there to be one binder, but it could be a combination of those that are identified in the Markush group. The USPTO has frequently objected to Markush claims as being directed to multiple inventions. The PTO has also tried to reject Markush claims under a judicially created doctrine of improper Markush groups. The CCPA has shot these down, but the PTO is undeterred. They are now rejecting Markush groups under yet another judicial doctrine. But well, as of right now, at least I can't find any support for this doctrine. But what's important to note is that what we see in the Idenix case and YS before that is that claims that are greedy and try to claim 10 to the 10th, 10 to the 100th structures by the use of Markush language are certainly at risk, and certainly with respect to the scope of enablement. So it's imperative that we include claims to a narrower scope with a reasonable number of alternatives and species, just as we were talking in the context of the antibody claims. It's also important for the specification to provide details to correlate structure and function where we are able to do so, as well as to provide as many examples as possible covering a diverse range of structurally variant species. As we should also look to provide clear explanations of methods of production and how efficacy is tested. So what we've seen in recent years is when the USPTO gets in its head that something should be a certain way, they don't typically stop their attempts until the courts have told them multiple times in multiple ways that they are wrong. Sometimes the USPTO even just slides new rules or interpretations either through to the examining court through internal memoranda, or by simply sliding it into the next version of the MPEP, as we've seen in multiple cases in the June release of the MPEP. Idenix smells of the next wave to come. The holding of a lack of enablement in Idenix gives the USPTO a much more effective tool to rein in the scope of Markush claims than relying upon some unidentified judicial doctrine. So it's certainly something that we see coming as the next wave. At this point, we thank you for your time and attention, and we'd be happy to answer a few questions. For those who do not, we will get to respond via email if you provide us with an email address. Of course, our emails are provided here on this slide, and we would be grateful to receive any feedback or comments that you might have and to answer any questions.
We'll also be sending a copy of the slides so that you would have it for your reference.
All right, well, we thank everybody for your attendance, and we hope to see you at some future time and event. Thank you very much, and this concludes our presentation.